In this lecture, we'll talk about development of transgenic plants with improved nutritional quality. We'll talk about the development of golden rice. So why we developed golden rice actually? Rice is a staple food and is consumed particularly in Asia and other countries also. It has been cultivated for over 10,000 years. And rice provides as much as 80% or more of the daily calorie intakes of 3 billion people, which is half the world population. So, in order to provide provitamin A to third world, particularly in developing countries where malnutrition and vitamin A deficiency are very common, so that's why rice was selected for. Rice, particularly generally consumed in its milled form with the outer layers removed. The main reason for milling is to remove the oil-rich alluvion layer, which turns rancid upon storage. It- Rice is milled just to improve the storage of the rice so it can be stored for a long period of time. But by removing alluvion, as a result, the edible part of the rice grain consists of the endosperm filled with starch granules and protein bodies, but it lacks several essential nutrients such as carotenoids exhibiting provitamin A activity. And vitamin A deficiency is a serious health problem in at least 26 countries in Asia, Africa, and Latin America. So this Golden Rice project was started uh, by Ingo Potares of the Swiss Federal Institute of Technology and Peter Bayer of the University of Freiburg. So they developed the golden rice. So golden rice is a variety of Rhizosphaera rice produced through genetic engineering of two biosynthesized beta carotene, a precursor of vitamin A in the edible part of the rice, that is endosperm of the rice. So this golden rice was developed by through genetic engineering by introducing beta carotene synthetic pathway in the rice endosperm. As you know, vitamin E deficiency can cause blindness among children and may even lead to that. According to WHO, dietary vitamin E deficiency or VAD causes some 250,000 to 500,000 children to go blind each year as a result of xerophthalmia and keratomalacia. The US recommended daily allowance for vitamin A is 400 microgram retinol per day for children aged 1 to 3 years and 800 microgram for the adults. Vitamin E deficiency often occurs where rice is a staple food since rice grain doesn't contain provitamin A or beta carotene. Rice produces so vitamin E deficiency is a big issue in developing countries in Asia, particularly in Asia, Africa, and South America. So rice produces beta carotene in the leaves, but not in the grains. Since rice is a staple food, so there is a deficiency of vitamin A in developing countries. So rice is consumed quite a lot in many countries. So, but rice produces beta carotene in the leaves, but not in the grains, where the biosynth pathway is turned off during the plant development. So the beta carotene is there in the leaves or of the rice plant, but not in the grain. So now the scientists introduced the whole beta carotene synthesis pathway in the rice endosperm to generate a transgenic golden rice. So by doing this, the resulting transgenic rice golden contain good quantities beta carotene, which gives the grain a golden color because of the beta carotene. That's why we call it golden rice. So genetic engineering was the only way to produce golden rice. Breeding was not possible because there is no rice germplasm capable of synthesizing carotenoid in the germplasm available. So breeding was not the option. So only genetic engineering could develop a golden rice. Transgenic approach has become feasible during recent years due to two reasons: the rapid progress in the development of rice transformation technology through biolistic method as well as using agrobacterium. The availability of almost complete molecular elucidation of the carotenoid biosynthesis pathway. So we now know the complete carotenoid biosynthesis pathway where the enzymes are involved. So it's all known now. In numerous bacteria and plants, which provide ample choice of bacterial genes and the plant cDNA to select from. And the advantage is that growers can reuse their seed as they please as compared to the transgenic plant which are developed by Monsanto. Golden rice offer people in developing countries a valuable and affordable choice in the fight against the malnutrition. It was a study of the boiled golden rice fed to volunteers concluded that golden rice is effectively converted into vitamin A in human. And a 2012 study of golden rice that fed 68 children aged 6 to 8 and concluded that the golden rice was as good as vitamin A supplements and better than the natural beta carotene in the spinach. So these studies confirm that golden rice can be used to provide sufficient amount of beta carotene to the children's. So now we'll talk about how the golden rice was developed. Biosynthetic pathway provitamin A is a continuation of lycopene pathway. Immature rice endosperm is capable of synthesizing geranyl diphosphate, that is GGDP, but subsequent stages of pathways are not expressed in the tissue. The subsequent enzymes were not available in the not expressed in the endosperm of the rice. Hence, the exogenous LYC genes has a transit peptide sequence attached to targeted to the plasmid where geranyl phosphate formation occurs. So this is a pathway for the synthesis of beta carotene, joining of two geranyl diphosphate GGTP molecule to form the precursor phytoene. The conversion of phytoene into beta carotene requires three additional enzyme activities, that is phytoene desaturase, beta carotene desaturase, and lycopene B cyclase. So this is the whole pathway of producing beta carotene. Cereal grains such as rice accumulate GGDP. Okay, so this step is there in the present in the endosperm of the rice. So geranyl diphosphate gets accumulated in the rice endosperm, but lack the subsequent enzyme in the pathway. All these enzymes are missing in the rice endosperm. So that's why the beta carotene is not synthesized in the wild type rice endosperm. So now to produce beta carotene in the rice endosperm, we need all these three genes to complete the carotene biosynthesis pathway. So the addition of three genes in the rice genome will complete the biosynthetic pathways. Phytogen synthesis gene is derived from the daffodil was used, which was fused to the rice endosperm with glutathione GT1 promoter. Phytogen synthase is a transferase enzyme involved in the biosynthesis carotenoid. It catalyzes the conversion of geranyl pyrophosphate to phytoene. Three steps required to convert phytoene to beta carotene: phytoene desaturase and carotene desaturase to introduce double bond to form lycopene. So lycopene cyclase is taken from soil bacteria Erwinia iridovora from rings to form rings in the beta carotene in the endosperm. Bacterial carotene desaturase capable of introducing all four double bonds can be substituted for the phytoene desaturase and carotene desaturase. So these enzymes can be introduced in the rice endosperm to make beta carotenoid in the rice endosperm. So now the manipulation of golden rice, or you can say to produce the golden rice, will require the introduction of three genes: phytogen synthase, carotene desaturase, and lycopene beta cyclase. So daffodil PSY gene rice gluten promoter constructs are inserted into the vector PZPSC along with bacterial carotene desaturase gene controlled by 35S promoter, as you can see in this one. PSY gene and CRT1 gene in this PZPSC. So both enzymes target to the plastid. Okay, so the plastid or the site of GGDP synthase. PSY gene by its own transit peptide and the CRT1 gene by the fusion to a P-ribulose 5,1,5 bisphosphate, carboxylase or oxygenase small subunit transit peptide sequence. Here you can see it here. This one, the CRT1 and PS5 gene. The lycopene beta cyclase gene with a functional transit peptide was inserted in vector PZL CYH here in this picture, as you can see, LCY gene under the rice endosperm specific glutealin promoter along with hygromycin resistance gene marker. Here in this LCY gene here in this construct. Another construct was the three vector constructed PB1 is used in a single transformation. This PB1 which has all CRT1, PSY, all three genes, whereas PZPS and PZLCYH are used in co-transformation. And here is the left border. So all these genes construct was put in between left border and the right border of the tDNA. Here in all these LB and right border, left border and the right border here, left border and the right border in both. So these now 500. So these plasmids were these constructs were constructed. These three vectors were then spliced into tDNA vector for transformation experiments, and they were using agrobacterium mediated DNA transfer. 500 pre-culture immature embryos were inoculated with an agrobacterium mixture of these plasmids, and the co-transfer plants were analyzed by southern hybridization to see whether these genes have been introduced into the rice genome. To determine the formation of beta-carotene, mature seeds from the transform lines and control plants were air-dried, dehusked, and polished with the emery paper. The color of the transform was observed. The hygromycin resistant plants were analyzed by southern hybridization for the presence of PSY and CRTL1 genes. So you can confirm whether the hygromycin resistant is the selection marker. So hygromycin resistant plants were analyzed by southern hybridization to see whether these genes have been introduced into the rice genome. The endosperm of these plant seeds were isolated and appeared yellow, indicating carotene. So once after observing the endosperm, if we see the yellow color, it means it is the carotenoid production is happening in the rice endosperm. And the high performance liquid chromatography HPLC analysis revealed the presence of beta carotene in the transgenic endosperm. So you can further confirm the presence of beta carotene in the rice endosperm by HPLC. So this is a slide to show you the beta carotene pathway in the plants, particularly in the rice. So geranyl diphosphate is converted to phytoin by phytoin synthase, and then phytoin desaturate carotene says convert phytoin to lycopene, and lycopene is then converted by lycopene beta cyclase into beta carotene. But in the rice case of the rice, all these three enzymes are missing, and now all these three enzymes by using tools of genetic engineering has been introduced now in the golden rice. So this is how it works actually. The geranyl diphosphate, which is accumulated in the wild rice endosperm, is now converted by phytoin synthesis, which is obtained from daffodil gene, and then phytoin synthesis, phytoin is produced. Phytoin desaturase and carotene desaturase is convert the phytoin into lycopene, and then lycopene is converted by lycopene beta cyclase from daffodil gene into beta carotene, which is a vitamin E precursor, and we get the golden rice. So this is how the genetic engineering has solved the problem of producing beta carotene in the rice endosperm now by using genetic engineering. In case of one transgenic line, beta carotene content was as high as 85% of total carotenoid present in the grain. One explanation is that the enzyme downstream along the pathway such as lycopene, cyclase and alpha beta carotene are still being produced in non-transformed rice endosperm, while PSY and phytoin desaturated are not. So this may be the reason that we got in few cases in one transcript line quite a high content of beta carotene. So this is again the same the whole pathway. So for after creating golden rice, there some more improvements were made to obtain better yield of beta carotene in the golden rice. So in one case, PM phosphomanose isomerase or mannose 6 phosphate isomerase mannose selection gene was substituted to avoid antibiotic selection with hygromycin resistant gene. In the first earlier hygromycin resistant gene was used for selection. So instead of that, mannose 6 phosphate isomerase gene was used to basically to avoid antibiotic selection. The golden rice trait was genetically engineered into indica rice cultivars. Indica rice is consumed by 90% of the Asian population, whereas the original golden rice is produced using japonica variety. So now they introduce the whole beta carotene synthesis pathway in the indica rice also. Subsequent research indicated that lycopene beta cyclase transgene was not required to produce beta carotene in the endosperm. So you can replace this gene also. Further work by Syngenta to optimize beta-carotene production showed that the daffodil phytoin synthesis was rate limiting. The enzyme phytoin synthesis which was obtained from daffodil was rate limiting, and PSY gene from maize was much more effective, which resulted in greater accumulation of total carotene and the beta-carotene. So after trying with PSY genes from different sources, it turned out that the maize and the rice gene gave the best results. So this was another improvement made. So this was instead of daffodil, now the maize PSY gene was used to create the golden rice version 2. So by using maize PSY gene in the golden rice lines was obtained that accumulated up to 37 microgram per gram carotene, of which 31 microgram was beta carotene as compared to the first generation golden rice, that is GR1, where only 1.6 microgram per gram was obtained. So you can see the yield. In case of golden rice version 1, only 1.6 microgram per gram of beta carotenoids was obtained. But in the golden rice 2, while using PSY gene from the maize, there was very good yield of beta carotene, that is 37 microgram per gram of carotenoid was obtained in golden rice version 2. So this increases the content of beta carotene in the rice endosperm. So basically the transformation of rice with the construct PSY resulted in 23-fold increase in carotene compared with the original golden rice and has been named golden rice 2. So this is the construct actually. To consider golden rice to the phytoin synthesis gene, PSY gene from maize and the carotene desaturase gene from the irvine was inserted in the rice. So here it is the PSY gene. So PSY gene from the maize, and here is the CRT1 gene. So this golden rice 2 produce more amount of beta carotene as compared to the version 1. In 2016, for India finds that the newer golden rice would reduce the burden of vitamin E deficiency in India by 5 to 54%. Although some beta carotene is disturbed during cooking and not all of it is absorbed into the body, the level of beta carotene in the golden rice too is comfortably enough to prevent vitamin E deficiency in the people eating ordinary amount of rice. So still even after cooking, there will be sufficient amount of beta carotene which will be enough to control the vitamin E deficiency in India. So this slide is actually just to show that if the impact of vitamin A deficiency in children. So, most importantly, it can lead to impaired vision. It reduces immune response, impaired skeletal growth. So all these, so vitamin deficiency can all these problems actually in children. So now these all these issues can be sorted out by consuming golden rice. So this slide actually shows the pictures of all the three rice. So this is the wild type rice variety which is white in color. This is golden rice version 1 which has low amount of beta carotenoid, and this is a GR2 golden rice version 2 which is by using maize PSY gene. So now this has more amount of beta carotenoid. So this golden rice version 2 is very much better and is to control the VAD. So what are the advantages of having a golden rice? Golden rice give more quantity of vitamin A, easy distribution when released to needy, cheaper option to supply vitamin A requirement compared to other supplementary measures, sustainable option as once released for common cultivation can be cultivated every growing season by farmers, saved seed, therefore no need of yearly budgetary investment for distribution. So once these seeds are distributed to farmers, they can save those seeds for the for further growth. So they don't have to keep on buying the seeds again and again. So it will be very cheap for the farmers to grow golden rice. But the green, some organization like particular GMO and others, they oppose the introduction of golden rice in the market. So it is argued that by promoting golden rice, we encourage a diet based on one staple rather than increase in access to many vitamin-rich food plants. So these plants would address a wide variety of micronutrient deficiency, not just VAT. G crops including golden rice have the potential to cause allergic reaction. So this is one issue GMO always raises. Supply doesn't provide substantial quantity as recommended by a daily intake. So these are the some of the issues for the golden rice. Environmental issues can be there like loss of biodiversity, may become a gregarious weed and endanger the existence of natural rice plants. Genetic contamination of natural global staple food. So this is another issue. Some people prefer to cultivate and eat only white rice based on traditional values and spiritual beliefs. So people may not opt for golden rice. And the financial interest can be there. The majority patents for genetically engineered plants are held by a few large multinational companies. So it's in their financial interest, not the farmers and the public. So these are some of the issues not only for the golden rice but for other transgenic plants. So this is this slide just to show you the yield of beta carotene in the rice endosperm. So golden rice version 1 accumulated around 1.6 microgram per gram of beta carotene in the grain. So golden rice version 2 produced around 31 microgram per gram of beta carotene. So this shows that golden rice 2 is quite good actually for for human consumption in developing countries. So this project, so this work was patented also. So this slide just to show you the current scenario, how actually uh, is it being consumed or it is just still in not accepted by the people. So the block, so this is the current scenario. The block on many scientists which approve this golden rice. They say that block on GM rice has cost millions of lives and led to child blindness. Eco groups and global treaty blamed for delay in supply of vitamin A enriched golden rice. As I showed you in an earlier slide, there GMO and so many other organizations oppose the introduction of golden rice in the market. But that has led to it has cost millions of lives and led to child blindness because they were children in the developing countries are not able to get sufficient amount of vitamin A actually. So many people, many millions of lives have been lost till now. So in particular, the international regulation have been blamed for delaying the approval of a food that could have helped save millions of lives this century. This claim is made in a new investigation of the controversy surrounding the development of golden by a team of international scientists. So they say that delaying the approval of the golden rice is causing a lot of death actually in poor developing countries. But in some developed countries, this has been accepted. Golden rice has been approved for consumption, actually for human consumption. The United States Food and Drug Administration (FDA) has approved golden rice, making the third positive international assessment for the genetically engineering by fortified crop. Previously, Food Standard Australia, New Zealand, and Health Canada gave golden rice the stamp of approval in Feb and March 2018, respectively. So these countries are already consuming golden rice. New Zealand, Australia, New Zealand, Canada, and USA. And in 2020, in January 2020, Newton's golden rice, a genetically modified amaranth crop, has passed a rigorous biosafety assessment in Philippines, where it may soon be distributed to combat the country widespread vitamin A deficiency. So this golden rice now is already being consumed in USA, Australia, New Zealand, Canada, and now Philippines has already approved it. So okay, so these few countries have already approved consumption of golden rice. So more and more countries needs to approve the marketing, production and marketing of golden rice so that they can combat the vitamin D deficiency in their children and save millions of lives every year.